Good day, this is Professor Will, CBMD PhD. Today is Tuesday, June 9th, 2020. It is 4:55 and 58 seconds a.m. Eastern Standard Time. And as you're about to see, we're going to previous research dates. So let's get started here. Today I'd like to make a very special announcement. Something special I've learned over the years of collaborating with my fellow PC techers. And that is, let's be part of the solution rather than part of the problem. Well, I hope we can all learn our life lessons. So let's get started here on this reading. Evolution of pandemic coronavirus outlines path from animals to humans. The virus's ability to change makes it likely that new human coronaviruses will arise. Date May 29, 2020. Source Duke University Medical Center. Summary. A team of scientists studying the origin of SARS-CoV-2, the virus that has caused the COVID-19 pandemic, found that it was especially well suited to jump from animals to humans by shape-shifting as it gained the ability to infect human cells. Researchers confirmed that the closest relative of the virus was a coronavirus that infects bats, but that virus's ability to infect humans was gained through exchanging a critical gene fragment from a coronavirus that infects a scaly mammal called a pangolin, which made it possible for the virus to infect humans. The researchers report that this jump from species to species was the result of the virus' ability to bind to host cells through alterations in its genetic material. By analogy, it is as if the virus retooled the key that enables it to unlock a host cell's door, in this case a human cell. In the case of SARS-CoV-2, the key is a spike protein found on the surface of the virus. Coronaviruses use this protein to attach to the cells and infect them, very much like the original SARS that jumped from bats to civets, or MERS that went from bats to dromedary camels, and then to humans. The progenitor of the pandemic coronavirus underwent evolutionary changes in its genetic material that enabled it to eventually infect humans. Researchers tracing the virus evolutionary pathway will help deter future pandemics arising from the virus and possibly guide vaccine research. The researchers found that typical pangolin coronaviruses are too different from SARS-CoV-2 for them to have directly caused a human pandemic. However, they do contain a receptor binding site, a part of the spike protein necessary to bind to the cell membrane, that is important for human infection. This binding site makes it possible to affix to a cell surface protein that is abundant on human respiratory and intestinal epithelial cells, endothelial cell, and kidney cells, among others. While the viral ancestor in the bat is the most closely related coronavirus to SARS-CoV-2, its binding site is very different, and on its own cannot efficiently infect human cells. SARS-CoV-2 appears to be a hybrid between bat and pangolin viruses to obtain the key necessary receptor binding site for human infection. There are regions of the virus with a very high degree of similarity of amino acid sequence among divergent coronaviruses that infect humans, bats and pangolins, suggesting that these viruses are under similar host selection and may have made the ancestor of SARS-CoV-2 able to readily jump from these animals to humans. People had already looked at the coronavirus sequences sampled from pangolins that we discussed in our paper. However, the scientific community was still divided on whether they played a role in the evolution of SARS-CoV-2. In our study, we demonstrated that indeed, SARS-CoV-2 has a rich evolutionary history that included a reshuffling of genetic material between bat and pangolin coronavirus before it acquired its ability to jump to humans. Hospitalized COVID-19 patients with diabetes represent more than 20% of ICU population. Date June 5, 2020. Source Endocrine Society. Summary. New guidance offers glucose management strategies that limit direct contact and minimize clinicians' risk. Hospitalized patients with COVID-19 and diabetes need to receive glucose-lowering therapy, in addition to other complex medical management as a way of minimizing risks for complications and death. However, appropriate glycemic management, including bedside glucose monitoring and insulin administration, requires intensive patient interactions and puts clinicians at risk. This manuscript provides guidance for healthcare providers caring for patients hospitalized for COVID-19 who also have a prior history of diabetes or who have high blood sugar levels at this time of hospitalization. These healthcare providers are at risk for contracting COVID-19, and while glycemic management in the hospital improves patient outcomes, it also intensifies the amount of time with direct patient contact. Clinicians may limit their risk of exposure by minimizing the use of IV insulin infusions and using remote glucose monitoring devices and non-insulin therapies when possible. Diabetes self-management by selected patients who are knowledgeable and capable of this in the hospital also can be considered as a way of limiting direct patient interactions. Clinicians should be aware that some medications used in treating COVID-19 patients, including glucocorticoids and hydroxychloroquine, can affect blood glucose levels. Could the blood of COVID-19 patients be used to predict disease progression? Study discovers differences in blood samples of severe and mild cases. Date June 5, 2020. Source University of Berlin. Summary. Researchers have identified 27 proteins which are present at different levels in the blood COVID-19 patients, depending on the severity of their symptoms. These biomarker profiles could be used to predict disease progression and make it easier for doctors to decide which type of treatment to use. People respond very differently to infection with the novel coronavirus SARS-CoV-2. While some patients develop no symptoms at all, others will develop severe disease and may even die. For this reason, there is an urgent need for biomarkers, quantifiable biological characteristics, which could provide a reliable means of predicting disease progression and severity. Researchers use state-of-the-art analytical techniques to rapidly determine the levels of various proteins in the blood plasma. This approach enabled the researchers to identify various protein biomarkers in the blood plasma of patients with COVID-19 which were linked to the severity of their disease. The researchers developed a precise high-throughput mass spectrometry platform capable of analyzing the patient's proteomes, the compendium of proteins found in biological material, at a rate of 180 samples per day. Using this technology, the team analyzed blood plasma samples from 31 men and women who were receiving treatment at Kuwait for COVID-19 of varying degrees of severity. The researchers were able to identify 27 proteins in the blood which varied in quantity depending on disease severity. The researchers then validated these molecular signatures by analyzing samples from another group of 17 COVID-19 patients and 15 healthy people. Protein expression signatures were able to precisely classify patients according to the World Health Organization's coding criteria for COVID-19. These results lay the foundations for two very different applications. One possible future use would be for disease prognosis. An early blood test would enable the treating physician to predict whether or not a patient with COVID-19 will develop severe symptoms. This could potentially save lives. The sooner physicians know which patients will require intensive care, the faster they can make use of the available treatment option. In order to get closer to this goal, the researchers will now study how the biomarker signatures change over the course of the disease. Another possible future use would be as in-hospital diagnostic tests, which could provide clarity regarding a patient's condition, regardless of how they themselves describe it. In some cases, a patient's symptoms do not appear to provide an accurate picture of their true health state. An objective evaluation based on their biomarker profile could be extremely valuable in this regard. The research team now plan to test their new method in a larger number of patients, in the hope of getting closer to developing a diagnostic test. Changes in the protein profile. Some of the 27 proteins which were found to predict the severity of COVID-19 had not previously been linked to an immune response. However, the biomarkers identified by the researchers also included clotting factors and regulators of inflammation. Some of these proteins act as interleukin-6 IL-6 at a molecular level. IL-6 is a protein which is known to cause inflammation, and which, according to preliminary studies, is associated with severe COVID-19 symptoms. A number of the biomarkers identified as part of this study might therefore be suitable targets for treatment. Applying symptom tracking to COVID-19 outpatient care using famotidine. Date June 5, 2020. Source: Cold Spring Harbor Laboratory. Summary: a patient-reported symptom tracking method used for patients with cancer has now been adapted for patients with COVID-19. The first clinical case series showed that famotidine may help COVID-19 patients with mild to moderately severe symptoms. The methodology is suitable for testing drugs in patients well enough to be managed at home, and allows the recruitment of diverse subjects via community-based on health organizations and individual practitioners. The experience of a patient at one point in time is very valuable, but learning about the change in their experience over time is even more important. Change indicates if the patient's condition is getting better or worse. A graded symptom scores enables the physician and the patient to track symptoms using numbers. Observing that for COVID-19 most symptomatic people do not require hospitalization, researchers and colleagues developed a 4-point scale for 6 common COVID-19 associated symptoms that patients score every day. Researchers supply how the scale can help track the course of a patient's disease. For example, you may call up your doctor and say, "I have headaches and shortness of breath, and am only able to do the basics of self-care," which would be grade 3 symptoms. If you still had symptoms 2 days later, but are now able to do light work, these symptoms would now be scored as grade 2. This approach makes it very easy for you and your doctor to document that your symptoms are improving. value of this approach from a research perspective is that experiences from many patients become comparable and can be pooled for analysis. If a drug speeds recovery, then most patients will report more rapid improvement of symptoms. These surrogate measures are very common and the best way to assess what's going on in the outpatient. We are actually pretty confident. We think there'll be another surge, but we think we'll be much better prepared and think that hopefully lives will be saved. Good day, this is Professor Will, CBMD PhD on infectious disease, microbiology, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.